Madam, audible? Hello? Hello? ಚೈತ್ರ ಮೇಡಮ್ ಅವರ ಹಲೋ. ಮೇಡಮ್, can you start now? ವಿದ್ಯಾರ್ಥಿಗಳ ತಮ್ಮಲ್ಲಿ ಒಂದು ಮನವಿ. ತಾಂತ್ರಿಕ ದೋಷದ ಕಾರಣದಿಂದ ಕಾರ್ಯಕ್ರಮವು ಸ್ವಲ್ಪ ವಿಳಂಬವಾಗಿದೆ. ಇನ್ನು ಕೆಲವೇ ಕ್ಷಣಗಳಲ್ಲಿ ಕ್ಲಾಸಸ್ ಶುರು ಆಗುತ್ತೆ. ಹಲೋ. 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 ಹಲೋ.
ಸಕ್ಕೆ ನಾನು ಅನ್ಮ್ಯೂಟ್ ಮಾಡಕ್ ಆಗ್ತಾ ಇಲ್ಲ ಅದ್ರಲ್ಲಿ. ಹೌದಾ? ಬಟ್ ನನಗೆ ಅನ್ಮ್ಯೂಟ್ ಮಾಡಕ್ ಆಗ್ತಾ ಇಲ್ಲ ಇದ್ರಲ್ಲಿ. ನಾನು ಲಾಗಿನ್ ಆಗಿದ್ದೀನಲ್ವಾ? ಲಾಗಿನ್ ತೋರಿಸ್ತಾ ಇದೆ ಅಲ್ವಾ? ಬಟ್ ಇದ್ರಲ್ಲ ಯು ಆರ್ ಮ್ಯೂಟೆಡ್ ಅಂಡ್ ಕಾಂಟ್ ಅನ್ಮ್ಯೂಟ್ ಯುವರ್ಸ್ ಅಂತ ಬರ್ತಾ ಇದೆ. ನೀವು ವಾಟ್ಸ್ಆಪ್ ಅಲ್ಲಿ ಲಿಂಕ್ ಕಳ್ಸಿದ್ರಲ್ಲ, ಡೈರೆಕ್ಟ್ ಆಗಿ ಅದ್ರಲ್ಲಿ ಕ್ಲಿಕ್ ಮಾಡಿ ಜಾಯ್ನ್ ಆದೆ. ಅಲ್ಲ, ಲ್ಯಾಪ್ಟಾಪ್ ಅಲ್ಲೇ ಡೈರೆಕ್ಟ್ ಆಗಿ, ಅಂದ್ರೆ ಲ್ಯಾಪ್ಟಾಪ್ ಅಲ್ಲಿ ವಾಟ್ಸ್ಆಪ್ ಓಪನ್ ಮಾಡಿ ಡೈರೆಕ್ಟ್ ಆಗಿ, ಅಂದ್ರೆ ಸಿಸ್ಕೋ ವೆಬೆಕ್ಸ್ ಇತ್ತು ಲ್ಯಾಪ್ಟಾಪ್ ಅಲ್ಲಿ, ಡೈರೆಕ್ಟ್ ಆಗಿ ಜಾಯ್ನ್ ಆಗಿ. ಹಾ ಹಾ, ಓಕೆ.
ಹಲೋ. 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 ಹಲೋ ಮೇಡಮ್. ಹಲೋ.
ಹಲೋ ಸರ್, ಕೇಳಿಸ್ತಿದೆಯಲ್ಲ?
ಮೇಡಮ್ ಕೇಳಿಸ್ತಿದೆ ಇವಾಗ? ಹಲೋ 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 ಮೇಡಮ್ ಇಯರ್ಫೋನ್ ಕರೆಕ್ಟ್ ಆಗಿತ್ತು ಮೇಡಮ್. ಕೆಳಕ್ ಹೋದ್ರೆ ಕರೆಯನ್ನ. ಹಲೋ
ಹಲೋ
ಮೇಡಮ್ ಕೇಳಿಸ್ತಿದ್ಯಾ?
ಹಲೋ ಕೇಳಿಸ್ತಿಲ್ಲ ಅಲ್ವಾ ನಿಮಗೆ ನಾನು ಮಾತಾಡ್ತಿರೋದು? ಹಾ. ಹಾ ಸರ್, ಸಿಸ್ಟಮ್ ಅಲ್ಲೇ ಎಲ್ಲ ಅಂದ್ರೆ PPT ಶೇರ್ ಮಾಡಿದೀನಿ ತಾನೇ, ಕಾಣಿಸ್ತಿದೆ ತಾನೆ? ಬಟ್ ನನ್ನ ವಾಯ್ಸ್ ಕೇಳಿಸ್ತಾ ಇಲ್ವಾ ನಿಮಗೆ? ಹಾ.
ಇವಾಗ ಮಾತಾಡಿ ಮೇಡಮ್. ಹಲೋ
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 ಹಲೋ.
ಕಟ್ ಮಾಡ್ತಿದ್ದೀರಿ ಒಂದ್ ನಿಮಿಷ.
ಹಾ.
ಇವಾಗ ಕೇಳಿಸ್ತಿದ್ಯಾ ಮೇಡಮ್? ಹಲೋ ಏನ್
ಹಲೋ. ಹಾ ಸರ್. ಐ ಥಿಂಕ್ ಬರ್ತಾ ಇದೆ ಅನ್ಸುತ್ತೆ ಆಡಿಯೋ. ಹಲೋ.
ಮೇಡಮ್ ಕೇಳಿಸ್ತಿದ್ಯಾ?
ಹಲೋ ಹಲೋ ಹಲೋ.
ಅದಕ್ಕೆ ಕೇಳೋದು. ಕಟ್ ಮಾಡಿದ್ರೆ ಅಲ್ಲಿ ಕೇಳಿಸೋದು. ನಾನು ಹೇಳಿದೆ ಇಯರ್ಫೋನ್ ಕರೆಕ್ಟ್ ಆಗಿ ಎಲ್ಲ ಇದೈತೆ.
ಹಲೋ.
ಮೇಡಮ್,
ಹಾ ಸರ್, ಹೇಳಿ ಸರ್.
ನನ್ನ ವಾಯ್ಸ್ ಸಿಸ್ಟಮ್ ಅಲ್ಲಿ ಕೇಳ್ತಾ ಇದೆಯಾ ಮೇಡಮ್?
ಸಿಸ್ಟಮ್ ಅಲ್ಲಿ ಸರ್, ಒಂದ್ ನಿಮಿಷ ಇರಿ. ಸಿಸ್ಟಮ್ ಅಲ್ಲಿ ಕೇಳಿಸ್ತಾ ಇಲ್ಲ ಸರ್, ಫೋನ್ ಅಲ್ಲಿ ಅಷ್ಟೇ ಕೇಳಿಸ್ತಾ ಇಲ್ಲ. ಸಿಸ್ಟಮ್ ಅಲ್ಲಿ ನಾನು ಅನ್ಮ್ಯೂಟ್ ಮಾಡಕ್ಕೆ ಆಗ್ತಾ ಇಲ್ಲ ಆಕ್ಚುಲಿ.
ಮೇಡಮ್, ಒಂದು ನಿಮಿಷ ನಿಮ್ಮ ವಿಡಿಯೋ ಆನ್ ಮಾಡಿ.
ಓಕೆ, ಒಂದ್ ನಿಮಿಷ.
ಏನ್ ಕರೆಕ್ಟ್?
ಹಲೋ ಮೇಡಮ್. ಹಲೋ ಮೇಡಮ್. ಹಲೋ ಹಲೋ ಹಲೋ. ವಾಯ್ಸ್ ಹಲೋ.
ಅವ್ರದಲ್ಲಿ ಅನ್ಮ್ಯೂಟ್ ಮಾಡಕ್ಕಾಯ್ತಾ ಇಲ್ಲ ಅಂತ.
ಸರ್, ವಾಯ್ಸ್ ನೀವು ಮಾತಾಡ್ತಿರೋದ್ ಕಾಣಿಸ್ತಿದೆ, ಬಟ್ ವಾಯ್ಸ್ ಕೇಳಿಸ್ತಾ ಇಲ್ಲ.
ಇವಾಗ ಇವಾಗ am I audible? ಕಾಲ್ ಮಾಡು.
ಹಲೋ
ಹಲೋ.
ಸರ್, ಇವಾಗ ಕೇಳಿಸ್ತಿದೆ ಅಲ್ವಾ?
ಹಾ.
ಹಾ, ಓಕೆ ಸರ್, ಓಕೆ.
ನನ್ನ ವಾಯ್ಸ್ ಬರ್ತಾ ಇದೆಯಾ?
ನಿಮ್ ವಾಯ್ಸ್ ಬರ್ತಾ ಇದೆ ಸರ್.
ಓಕೆ ಫೈನ್ ಸರ್.
ಐ ಥಿಂಕ್ ಇದರಲ್ಲಿ ಸ್ಪೀಕರ್ ವಾಲ್ಯೂಮ್ ಕಡಿಮೆ ಇದೆ ಅಂತ.
ಓಕೆ.
ಓಕೆ.
ನಮಸ್ತೆ ಮೇಡಮ್.
ಎಸ್ ಸರ್, ನಮಸ್ಕಾರ.
ಸಂತೋಷದಿಂದ ಕಾರ್ಯಕ್ರಮ ಸ್ವಲ್ಪ ಲೇಟ್ ಆಗಿ ಶುರುವಾಗ್ತಾ ಇದೆ. ಸೊ ಟುಡೇಸ್ resource person, I welcome to Chaitra.
Yes sir, thank you sir.
So madam currently working at Base Education Private Limited and also have teaching 6 years teaching experience, and she has published a paper in Journal of Material Chemistry and Royal Society of Chemistry titled Self Assembly of Chiral Nematodes. Have secured first rank in biodiversity and the role of medical plants in primary health care. So over to you, ma'am.
Yes sir, thank you sir. Hello students, good evening. I think ನನ್ನ ವಾಯ್ಸ್ ನನಗೆ ವಾಪಸ್ ಬರ್ತಿದೆ ಅಲ್ಲ?
ಎಕೋ ಬರ್ತಾ ಇದೆಯಾ?
ಹಾ ಸರ್.
ಅಲ್ಲಿ ಸ್ವಲ್ಪ ವಾಲ್ಯೂಮ್ ಕಂಟ್ರೋಲ್ ಮಾಡ್ಕೊಳ್ಳಿ ಮೇಡಮ್ ನೀವು ಅಲ್ಲಿ.
ಹಾ ಸರ್. ಹಲೋ.
ಹಾ ಹೇಳಿ ಮ್ಯಾಮ್.
ಒಂದ್ ನಿಮಿಷ ನನ್ನ ವಾಯ್ಸ್. ಹಲೋ.
ಹಾ ಹೇಳಿ ಮೇಡಮ್.
ಸರ್ ಎಕೋ ಕೇಳಿಸ್ತಾ ಇದೆಯಾ ವಾಯ್ಸ್?
ಇಲ್ಲ ಎಕೋ, ನಮ್ಗೇನು ಎಕೋ ಬರ್ತಾ ಇಲ್ಲ ಮೇಡಮ್ ಈ ಕಡೆ.
ಓಕೆ ಸರ್, ಹಂಗಾದ್ರೆ ಓಕೆ. ಓಕೆ ಸರ್, ಶುರು ಮಾಡಿ.
ಈಗ ಯಾರೋ ಒಬ್ರು ಸ್ಟೂಡೆಂಟ್ಸ್ ಕಳಿಸಿದ್ರೆ ವಾಯ್ಸ್ ನಾಟ್ ಕಮ್ ಸರ್ ಅಂತ. ಬಟ್
ಇವಾಗ ಕೇಳಿಸ್ತದೆ, ನಿಮ್ ವಾಯ್ಸ್ ಕೇಳಿಸ್ತದೆ ನನಗೆ, ನನ್ನ ವಾಯ್ಸ್ ಕೇಳಿಸ್ತದೆ.
ಕರೆಕ್ಟ್ ಇದೆ, ಆಡಿಬಲ್. ಪ್ಲೀಸ್ ಕ್ಯಾನ್ ಸ್ಟಾರ್ಟ್ ನೌ ಮ್ಯಾಮ್.
ಓಕೆ ಸರ್, ಥ್ಯಾಂಕ್ ಯು.
ಓಕೆ.
Okay, hello students. In this class, we will be studying the important biomolecule, which is carbohydrate. So let us understand what exactly it is. So this is a picture, whatever I have shared here, which contains carbohydrates. So that's what our today's topic is. You know how much important carbohydrates for our body. And this is the structure of proteins. This is the primary structure, secondary structure, tertiary and quaternary structure. Proteins are also very important biomolecules for everyday life, as well vitamins. So you know that if vitamin deficiency is there, so many problems will come into picture. Like if you have deficiency of vitamin A, for example, night blindness will be caused. As well as if vitamin C deficiency is there, scurvy can occur. So similarly, we have DNA and RNA, which is the very important genetic material which is required. So those are also biomolecules. And this is the RNA structure, whatever I have shown. So all this slide, whatever I have put, they are the biomolecules. So basically, biomolecules are organic compounds, but they are not simple organic compounds like whatever you see, like simple alcohol, like methyl alcohol, ethyl alcohol, or alkenes, etc. So these are complex organic molecules or compounds which are very important or important constituents of living organisms which are present in different cells. So ಯಾಕೆ ಇದೆಲ್ಲ ಇಂಪಾರ್ಟೆಂಟ್ ಅಂದ್ರೆ they are responsible for growth. So if we have to grow or maintain our life and the ability to reproduce, everything is because of this biomolecules. they basically play a very important role. So these are the common biomolecules. So let us start in detail what exactly carbohydrates are. This is the definition, whatever I told. This is the hierarchy. Yes. So these are the general biomolecules. So let us start with carbohydrates. So these are produced by plants actually. So we completely, animals as well as human beings, we rely on plant sources. So which are the important carbohydrates? As soon as I ask, you would remember the glucose example, right? All of you would have seen whenever sports day takes place, generally glucose they would have kept, right? So that is an instant source of energy. So if you feel tired, you can consume glucose immediately. That will increase the blood sugar level, right? You'll be more active. See, as well starch, fructose, these are also carbohydrates. So basically the meaning carbohydrates is nothing but it is made up of carbon and water. Actually, this is the misconception. Previously, we considered this meaning. See here, it is made up of carbon, hydrogen, as well as oxygen. In what ratio? We will observe through the example. I have written here generally 1 is to 2 is to 1. Now let me take one example. The first example I'm taking is glucose. What is glucose? C6H12O6, right? It is the simplest. Simplest means just one monosaccharide unit will be there. So I can actually express glucose in this format C6H2O 6 times. If you multiply 6 inside, you would get this formula only. Now, one more example I'll take, that is sucrose. Again, if you take sucrose, you can also write this as C12H2O 11 times. So this is nothing but the format in which whatever I have written this formula is nothing but hydrates of carbon, right? So I can call this carbohydrate general formula is the CXH2O times. Okay, this is the general formula. But there is no rule everybody should satisfy this. Here, in this example, if you take x is equal to y, 6 and 6. But in this example, x is not equal to y. But again, they are fitting in this general format. So I can call, okay, these are hydrates of carbons. Okay. But later on, we will understand there are some carbohydrates which doesn't fit in that particular formula. So let me show that example for you. See, this is the molecular formula CH3COOH. This is acetic acid. It's a carboxylic acid. It is not a carbohydrate, right? Acid functional group is there. So this is called as carboxylic acid. But if you try to fit in that particular formula, let me try and show it to you. Here, you can take C. One second. C. You can take C 2 times and H 4 times and O2. Now I can represent it as C2 H2O 2 times. If you multiply this, I'm getting this only. This is not a carbohydrate, but still it is fitting in this formula. Whereas raffinose is a carbohydrate, but it doesn't fit into that formula. So basically, what is the understanding? The formula, general formula was given for carbohydrates, but some carbohydrates were accepting that format, whereas some were not accepting that. Whereas some other molecules which are not at all carbohydrates were able to represent in this format. So that gave a lot of confusion for people. That's why that is not a better definition of carbohydrates. Now we have a different carbohydrate definition. So I can call carbohydrates are polyhydroxyaldehydes or ketones, or which upon hydrolysis heat that particular molecules. Okay, I'll show that structures. Let us understand the basic meaning of carbohydrates. Uh, they're also called as saccharides. In Greek, actually, saccharon means sugar. So that's why they are called as saccharides also. Now, classification we will understand. So, classification of saccharides. They are classified into three types: monosaccharides, oligosaccharides, and polysaccharides. So, what are these monosaccharides? Basically, that is a carbohydrate which cannot be further hydrolyzed. Now, if you take glucose, you can't break it. That is the very simple unit, sugar unit. Okay. This is the general formula CnH2nOn for monosaccharides, where N can be approximately 3 to 7 or 3 to 9 also. Okay. This is the very simple unit. Approximately 20 monosaccharide units are known, or sugar units are known in nature. These are very important one: glucose, fructose, ribose, etc. So we will study in detail glucose today. Okay, I will teach you glucose structure. Whereas what are oligosaccharides? They are the carbohydrates which contain few monosaccharide units, 2 to 10. So as I have mentioned here, they can contain 2 to 10 monosaccharide units. Fine. Further, they'll be classified as disaccharides, trisaccharides, tetrasaccharides. For us, important one is disaccharides. For example, sucrose, if you break sucrose or upon hydrolysis, it gives glucose and fructose. Now, last one among this saccharides, polysaccharides. This is the general formula C6H10O5 enzymes. So polysaccharides means many sugar units will be joined. One of the example you can imagine is starch. Starch is made up of n number of glucose. You know the molecular formula of glucose, but it is alpha glucose. Structural difference will be there. Cellulose is made up of glucose, but it is beta glucose. Okay, so these are the polysaccharides. In the same manner, you have different classification. Before that, just the nomenclature I will show. If 3 carbon atoms are there in the sugar unit or monosaccharide, you call it as triose. Tri for number of carbon atoms and ose for sugar. And if aldehyde functional group is there, aldotriose, ketone is there, ketotriose. Like that, only 4 carbon atoms means tetrose, 5 means pentose, 6 is hexose, 7 is heptose. Okay. So one by one we will take later example. One more classification actually. So based on functional group, you have classification based on taste as well as solubility. Based on nature of reaction, you have one more time. Okay. Based on functional group, this is the actual modern definition I gave. Carbohydrates are nothing but optically active. So what is the meaning of this optically active? Means in nature, there are some organic compounds which have the ability to rotate polarized light in a different manner. So if they rotate light towards right hand side, they'll be called as dextrorotatory compounds or clockwise rotation you can call. If they rotate polarized light towards left hand side, they are called as levorotatory compounds. Okay, so you can call carbohydrates as optically active polyhydroxy means this is the functional group. Many OH group will be there. Aldehydes, it can be having CHO group or it can be having C double bond O group. So carbohydrates are nothing but optically active polyhydroxyaldehydes or ketones. Or there are bigger units like starch you can take. If you take starch and hydrolyze it, you will get glucose. N number of glucose. So that's why they are given, or the compounds which produce such units upon hydrolysis. Okay, that's the definition based on functional group. Now I'll show the structure you see. This is glucose, C6H12O6. Always the first carbon atom first you have to write, that contains the functional group, important prioritized functional group. Other alcoholic groups are here. Okay, this is the first carbon atom, consecutively 2, 3, 4, 5, 6. Fine. So it has aldehyde as well as alcohol. But the nature is different, alcoholic nature. Primary alcohol is there as well as secondary is there. So glucose, it has aldehyde as well as alcohol group. That's why it's called as polyhydroxyaldehyde. Similarly, one of the other important monosaccharide is fructose. Here, what is the changes? At the second position, there is a keto group, and in the first position, there is just CH2OH instead of CHO. Rest all is same. Okay. So fructose is also containing alcohol group, but other than aldehyde, it is having ketone. So it's polyhydroxy ketone. That's why this is the modern definition. Their polyhydroxyaldehydes are ketones. Now, based on taste and solubility, you have one more type, sugars and non-sugars. Sugars, they will be generally crystalline. You would have seen that sugar, whatever you use at home, right? Crystalline. They'll be sweet to taste. And also they'll be soluble in water. Take a glass of water and mix sugar, it will dissolve, right? Simple examples are glucose, fructose, all those. These are not sugars. Non-sugars are amorphous and tasteless. They'll be bland to taste. Basically, the monosaccharides which are having low molecular mass, they will be sweet in taste. Okay. And they'll be insoluble in water. So examples for that starch, cellulose, glycogen, you can remember. Polysaccharides basically. And the last type of classification, based on the nature of the reaction or reducing ability, you have reducing sugars and non-reducing sugars. Fine. So what is the basic meaning of this? We'll understand. So if you have studied already aldehydes and ketones chapter, if functional group aldehyde is present, they reduce spelling solutions and Tolling's reagent, right? So these reducing sugars reduce Spellings and Tolling's reagent. Basically, that aldehyde group will be converted into COOH group. So all monosaccharides are reducing. The basic idea is that monosaccharide will contain a open functional group. That aldehyde group is free for oxidation, then it is called as reducing sugar. Okay. Whereas disaccharides in which aldehydic and ketonic group is free, for example, if I consider a disaccharide, it is made up of 2 sugar units. Okay. At least one functional group will be open for oxidation. Other one will be involved in a linkage, glycosidic linkage. At least one functional group is open for oxidation. So the examples for that is maltose and lactose. Whereas one more disaccharide is there, sucrose, that I'll show non-reducing sugar. So these non-reducing sugars, if 2 units are there also, their functional groups are present like this. They are involved in the glycosidic linkage. So it's not open for oxidation. That's why they're called as non-reducing sugars. And the example for that is sucrose. Okay. And all polysaccharides are also non-reducing sugars because their functional group will be involved in the linkage. Okay. Now, preparation of glucose I'll show. Then we'll study the chemical properties. So there are 2 methods of preparation. One is lab method and commercial method. So this glucose is very abundant in nature. So it is found in grapes. Generally, in all the fruits, fructose will be there, but in grapes, more of glucose will be there. In honey, it is present. In human blood, it is present. That's why it has different names. It is also called as grape sugar because it is present in grapes and it is present in human blood. That's why it's also called as blood sugar. Then it is also present in corn. So it's called as corn sugar. And it is also called as dextrose because it can rotate the plane polarized light towards right hand side. That's why it's called as dextrose also. Okay. See? Okay. Now preparation. First I'll show you lab method, then we will understand the commercial. So laboratory method is from sucrose. Sucrose is also called as cane sugar. Okay, not only from sugar cane, from other substances also they can synthesize this sucrose. Now, sucrose formula I have written. Upon hydrolysis, it will give glucose plus fructose. What is the difference between glucose and fructose? They do have same molecular formula, right? But different functional group will be there. In glucose, you will have aldehyde group as functional group, whereas in fructose, there is keto group as the functional group. Okay, and on the aroma quota, I have written here acid. Generally, this reaction happens in the presence of acidic medium, right? And also, one more thing you can note down: this reaction happens in the presence of invertase enzyme, the enzyme which carries out this is invertase. And this will be dextrorotatory, this will be levorotatory, and the process is also called as inversion. Okay. And we want glucose, right? I'm explaining you the synthesis of glucose. So how can you separate fructose? That's the question, right? So now you see, actually fructose is more soluble in alcohol. Glucose is less soluble. So if you cool, glucose will get crystallized. So you can just do filtration and separate it. Okay? That's how you will synthesize in laboratory. Next one, commercial method. That is from starch. Starch, general formula, this is C6H10O5. If you hydrolyze it in the presence of again acidic medium and temperature and pressure, you can notice I'll get n number of glucose. So starch is basically a polymer, whereas glucose is a monomer. So if excess of sulfuric acid is there, you can add just chalk powder or CaCO3 and neutralize it. This is the commercial method. And you can crystallize glucose molecules and extract it. So today's important topic is structure of glucose only. What and all is present in the structure we will understand. So as I earlier told, it's an aldohexose. Why it is aldo? Because glucose contains aldehyde group. How it is called as hexose or why it is called as hexose? It has 6 carbon atoms and it is a sugar. That's why I'm ending it with OSC, aldohexose. It's also called as dextrose. Already I have told you, it rotates the light towards right hand side. That's why dextrose. Now we'll go to the structural elucidation. So IUPAC name of glucose is 2, 3, 4, 5, 6 pentahydroxyhexanal. How many OH groups are there? 5 OH groups are there. 5 OH groups. Whereas how many CHO group? 1 CHO group. Okay, that's why. Molecular formula this is. So first evidence how glucose structure is present we have done this test. Take glucose. Instead of again and again writing CHOH, I can write CHOH 4 times. Okay. Now, if I heat it with HI, hydrogen iodide, rigorously, it will get reduced to CH3, CH2, 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 CH3. Total 6 carbon atoms are only there. So here also I have written 6 carbon atoms. This is nothing but N-hexane. Okay. So this suggests, since everything is in one line after this reduction, you can understand that glucose contains the carbon atoms linked in the same chain, like straight chain. There is no branching as such. It's just straight chain compound. That's the first evidence. Like they try to understand what is the exact structure of glucose, right? Now, glucose reacts with hydroxylamine to form oxime, and it also reacts with hydrogen cyanide, that is HCN, to give cyanohydrin. Actually, in aldehydes and ketones chapter, you have studied how aldehydes and ketones or carbonyl compounds reacts with oxime. Oxime is nothing but, hydroxylamine, NH2OH to form oxide. How they react with HCl to give cyanohydrin. If they are reacting with this, it is understood that there is a carbonyl group present in the glucose. Earlier, they didn't know that they had aldehyde group. They knew that carbonyl group is present. By doing this reactions, the test of hydroxylamine and hydrogen cyanide, then they discovered the other one. Now you see, I'm taking glucose, treated with hydroxylamine. When glucose reacts with hydroxylamine, it forms oxide. Whenever CH double bond NOH, the structure is that you will call it as oxide. Since it is derivative of glucose, you are calling it as glucose oxide. Okay. Next, one more structure I will take. Again, take glucose and treat it with HCN. Again, H plus Cn minus. Here you have carbonyl group like this. Okay. Now you see, whenever carbonyl group is present, oxygen is more electronegative. You know that electronegativity difference will be there. Carbon get positive charge and oxygen get negative charge. For that negative charge, you will be attaching the H plus, whereas positive part, you will be attaching the Cn. That's how you are getting cyanohydrin. This is called as glucose cyanohydrin. This proved that glucose contains carbonyl group. Now, you know that glucose contains carbonyl group. Like aldehyde also contains carbonyl group. Ketone also contains carbonyl group. What it is? To understand that, let us take the next one. Glucose, when I treat with mild oxidizing agent like bromine water, the aldehyde group which is present gets oxidized. So you should know that aldehyde group oxidizes to acid, COOH. See now? So this is called as gluconic acid. This indicates that this is a mild oxidizing agent. If it is turning into COOH, the meaning is this should be aldehyde group only. So this reaction suggests there is aldehyde functional group. Now you know the definition of carbohydrates already. It just doesn't contain aldehyde group. It contains OH group also. Next further test. Now on oxidation, nitric acid. On oxidation with nitric acid, glucose is dicarboxylic acid. The CHO will also get oxidized because this is a strong oxidizing agent. This primary alcoholic group also converts into COOH. That's saccharic acid. Okay. Or else you started with gluconic acid, the earliest step, whatever we got now. Do oxidation with HNO3. Once again, this primary alcohol gets converted into acid. That proves that there is a presence of one primary alcohol in the glucose. Fine. Now, by next test, we can understand that different nature of alcoholic group. This should be primary alcohol. Otherwise, it is converting into COOH with strong oxidizing agent, which it's understood that it should be primary alcohol. Rest all, what is the nature? Let us understand. This indicates presence of primary alcoholic. Next, let me take acetic anhydride. All of you know the formula of acetic anhydride. This is the formula CH3CO twice O, acetic anhydride. It's a derivative of acetic acid basically. Take glucose, treat it with acetic anhydride. Now what happens? You know OH is also acidic in nature, no? How many OH groups are there in this carbon? Means totally 4 carbon atoms are there. Each carbon atom contains one OH group, no? Now, whenever you have acetic anhydride, I'll open and show the structure. This is the structure of acetic anhydride. Now, these hydrogens are acidic in nature. Okay. So, you will do the acylation. See, basically, the CH3COO minus will take away that H plus and go out as acetic acid, CH3COOH, and rest acyl group is there, no? CH3CO, that is getting substituted here. So, how many times? 4 times. That indicates there are 4 OH groups which are secondary. Secondary OH means the carbon should be secondary. It should be attached to 2 different carbon atoms. Now you observe, this indicates this glucose pentacetic. And also, you might get a doubt, how come one carbon contains one OH group or different carbon only different OH groups are there? Why not at the same carbon atom? But when you treat it with this acetic anhydride, right? You get pentacetate, no? This is a bulky group, right? If it is existing, that tells that it is stable molecule. That also tells that the carbon atom containing OH group should be different. Otherwise, stable structure won't be available. So this reaction confirms presence of 5 OH groups. Since it exists as a stable compound, the 5 OH group should be attached to different carbon atoms. Okay, and this is the open chain structure. See, CHO up and CH2OH down, and HOH you can write in this pattern. This is glucose. And already I showed you gluconic acid means up COOH will be there. Saccharic acid means up and down both COOH will be there. Rest all will be same. So all this is the structural elucidation. By this we understood that glucose is a polyhydroxy aldehyde group will be there as a functional alcohol group as well as aldehyde. So, and also glucose is named as D plus glucose. Why plus? It is dextrorotatory. It rotates the plate polarized light towards right hand side. Okay. And why capital D? Capital D represents the highest numbered chiral carbon atom contains OH on which side? Actually, this capital D, capital L notation is because of in glyceraldehyde, OH is on right hand side, we call it as D notation. If OH is on left hand side, you will call it as L. Okay. So capital D represents OH configuration, whereas small d, small l represent dextro and levo rotation. Right? That's what they have indicated here. And also there is this all naturally occurring carbohydrates are D form, whereas all naturally occurring amino acids will be L form. So this notation is also important to understand the structures. You cannot tell whether the compound is dextrorotatory or levorotatory by looking at it, but you can tell capital D or capital L. See, this is the simplest carbohydrate with aldehyde group, glyceraldehyde. OH is on right hand side, that's why you are calling it as capital D. Okay, we know that if it is dextrorotatory, you can write plus. Otherwise, you can't simply write. Okay, and this is levorotatory, and L, capital L represents OH on left hand. So this is the part which you can look for in glucose. Actually, we tell now you can have confusion how to tell whether it is capital D or capital L. OH is on right hand side, that's why capital D. OH left hand side, capital L. No, but here both the sides OH is there. How can you tell it is capital D? You should check the highest numbered chiral carbon atom. Chiral carbon atom is one which is attached to four different atoms or group of atoms. So this carbon, if you consider, that is the highest number. Numbering should be done from this end only. Why? Because aldehyde should be getting the highest purity. So this contains OH on right hand side. That's why you'll be calling it as D-glucose. Okay. And IUPAC name is 2, 3, 4, 5, 6 penta hydroxy hexane 1R. Okay, this is all the structure. So always you should take reference as glyceraldehyde only. Based on that only, Emil Fischer has given this notations. And the open chain structure is also given by Emil Fischer. But as I told here, open chain structure, there is also cyclic structure of glucose. When scientists try to conduct certain reactions, despite having aldehyde group, glucose will not answer those tests. For example, there is Skipp's test, reaction with ammonia, aldehyde with ammonia, and reaction with sodium bisulfite. All these reactions will not be answered surprisingly. Okay, so that proved that glucose cannot exist in only one structure. It might exist in another structure also, which is nothing but the cyclic structure. Okay, it can exist in two different crystalline structures named as alpha and beta. So, what is the difference between alpha and beta? I'll show you here. Structural pattern I'll show. This is the open chain. So during ring formation, the carbon atoms which are involved are C1 and C5. Between C1 and C5, there will be hemiacetal linkage. Now you see, there are two isomers. One is alpha D-glucose and another one is beta D-glucose. So what is the difference between alpha and beta? Now you check the first carbon atom. In this case, alpha, OH is on right hand side, whereas in this case, OH is on left hand side. So, if the carbon atom contains OH right hand side, you call it as alpha. Left hand side, it is beta. Or else you can also call the relationship between these two is anomeric, and these two are also called as anomers. What are anomers? The stereoisomers which differ in the configuration of first carbon atom. If any other carbon atom configuration is different, you should be calling it as epimers. Why these are called as anomers? Because the first carbon atom configuration is changed. Fine. These are the cyclic structure. So you should remember with what and what carbon that hemiacetal linkage will be there. It is C1 and C5. Now you see, this is one more thing. Harvard structure. You take the pyrite as the skeleton. In reference to that, you will write glucose structure, which is given by Harvard and also called as Harvard structure. Okay, you can write first carbon atom, second, 3, 4, 5, and the sixth, always sixth carbon atom should be on the top according to the IUPAC notation. Now you can know the hemiacetal structure. Based on that, whatever is right hand side, you can write that down. Whatever is left hand side, you can write that up. That becomes the Harvard structure. If one is the alpha form, in alpha form, OH will be down as on first carbon atom. In beta form, OH will be up. That's the only difference. Okay, that is the beta. So that skeleton, whatever I have written, that is pyrene. It's a cyclic organic compound with one oxygen atom and 5 carbon atoms in the ring. Count how many carbon atoms are there? 1, 2, 3, 4, 5. And one more oxygen atom. It looks like a hexagon. This is pyrene. Just we have taken its skeleton and written this. So the six-membered structure of glucose is also called as pyrenose because it is in analogy with the pyrene. So the first structure, whatever I have written is alpha D glucopyranose in reference to that pyrene you are telling, and the other one is beta D glucopyranose. So this is the definition I already told you. First carbon atom configuration is different means it is anomers. Check here. Here, first carbon atom. OH is down. And here OH is up. Epimers means other than first carbon atom, any configuration is changed. So similarly, you have fructose also, one of the other monosaccharide. This is present in most of the fruits. It is the sweetest of all the sugars. It's called as levulose because it is levorotatory. And how to prepare means same like glucose only. If you break or do hydrolysis of sucrose, you will get glucose as well as fructose. And molecular formula is same. So this is the structure. Up CH2OH, down also CH2OH. But on the second carbon atom, you should write ketone because keto group is not terminal like aldehyde. Just levorotatory and keto group. So this is polyhydroxyketone. So again, here also hemiacetal linkage takes place, but not between the first carbon atom and fifth. It is between the second carbon atom and the fifth carbon atom. This is the alpha, and this is beta. And this you won't call it as anomers because the second carbon atom's configuration has changed. So you will be calling it as epimers. So now we'll take the help of urine, a 5-membered cyclic compound, which has 4 carbon atoms and 1 oxygen atom. With that help, we will be writing the Harvard structure of fructose. And the name is called as fructofuranose. Just whatever is right side, you will be writing. Left side and right side, up and down, you will be writing. So this is all the structures of monosaccharides, importantly glucose and fructose. So this is all about the today's class. We have understood the monosaccharides. Hello, sir.
ಡಿಯರ್ ಸ್ಟೂಡೆಂಟ್ಸ್, ಏನಾದರೂ ಈ ಟಾಪಿಕ್ ಮೇಲೆ ಕ್ವಶನ್ಸ್ ಏನಾರ ಇದ್ರೆ ಮೇಡಮ್ ಇಲ್ಲೇ ಕೇಳ್ಬೋದು. ಮೇಡಮ್ ಯಾವುದೇ ಕ್ವಶನ್ಸ್ ಇಲ್ಲ. ಲೇಟರ್ ಆನ್ ಐ ಸೆಂಡ್ ಥ್ರೂ ಮೇಲ್ ಮ್ಯಾಮ್.
Yes, sir.
So today delivered a very good informative talk on carbohydrates.
Thank you.
So I thankful behalf of KST and myself. Thank you, ma'am.
Thank you, sir.
ಸೊ ಇವತ್ತಿನ ಒಂದು ತಾಂತ್ರಿಕ ದೋಷದಿಂದ ಕ್ಲಾಸ್ ಸ್ವಲ್ಪ ಲೇಟಾಗಿ ಶುರುವಾಯಿತು. ವಿದ್ಯಾರ್ಥಿಗಳಲ್ಲಿ ಒಂದು ವಿನಂತಿ ವಿಷಾದಿಸುತ್ತೇನೆ. ಸೊ ನಾಳೆ ಕೂಡ ಗ್ಲೋಬಲ್ ವಾರ್ಮಿಂಗ್ ಬಗ್ಗೆ ಡಾಕ್ಟರ್ ನಾಗೇಶ್ ಹೆಗಡೆ ಅವರು 4 ಗಂಟೆಗೆ ಕ್ಲಾಸ್ ತಗೋತಾ ಇದ್ದಾರೆ. ತಾವು ದಯವಿಟ್ಟು ಇಂದಿನಂತೆ ಹೆಚ್ಚಿನ ಸಂಖ್ಯೆಯಲ್ಲಿ ಹಾಜರಾಗಿ ಆ ಒಂದು ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮವನ್ನು ಯಶಸ್ವಿಗೊಳಿಸಿಕೊಡ್ಬೇಕೆಂದು ಮತ್ತೊಮ್ಮೆ ತಿಳಿಸುತ್ತಿದ್ದೇನೆ. ಓಕೆ, ಥ್ಯಾಂಕ್ ಯು ವರಡಾಲ್. ಥ್ಯಾಂಕ್ ಯು ಮ್ಯಾಮ್.
Thank you, sir.
ಓಕೆ, ಥ್ಯಾಂಕ್ ಯು.